Sure. If you want to start with your branded slide while you do the introduction, that's fine. We can switch between the two.
That's okay. We'll just go from yours. Yours
Okay.
is all loaded up.
Okay.
Good morning, everyone. We'll give it just a minute or two as people are still logging on, and then I'll introduce you to our speaker today. Good morning, everyone. Welcome to the April AIDS Clinical Conference. And it is my pleasure to introduce today's speaker, Dr. Paula Cannon, who is a distinguished professor at the Keck School of Medicine at the University of Southern California, who has a long-standing commitment to HIV cure research. She began her academic career at the University of Liverpool, studying microbiology and later receiving a PhD in genetics. And she did postdoctoral work at Harvard and the University of Oxford, studying HIV and gene therapy. She is currently on the Genome Engineering Committee of the American Society of Gene and Cell Therapy, and her lab focused on the application of gene and stem cell therapies to target HIV infection. She's going to speak to us today about gene therapy for HIV cure. Thank you so much, Dr. Cannon, for joining us.
Thank you, Shirisha. Well, good morning, everyone. So what I'm going to try and do is give you an overview of some recent developments in gene therapy for HIV cure. And I know many of you are clinicians or also work with HIV positive individuals. So you might be thinking, gene therapy? We're not doing that right now. So I hope I can make you at least believers in the potential. All right. So let's start off with a bit of background. So gene therapy, even though it's kind of new, has been sort of around for quite a long time. It's been sort of officially a good idea since 1972, when this sort of article appeared in Science, talking not just about the idea of gene therapy for human disease, but also, very early on about the sort of ethical problems that this might create. And then it took almost another 20 years before the first gene therapy trial actually kicked off. So this was in 1990 at NIH. The first approved gene therapy trial was for an immune deficiency SCID ADA. And in the 10 years after that initial trial, there was a great deal of enthusiasm, and you'll see the number of trials that were initiated worldwide really exploded, especially for cancer, which where the risk-benefit kind of equation was considered favorable. More recently, we definitely went through a phase where it was where people were less interested when these initial trials didn't, provide home run treatments. But more recently, interest has really picked up again, in particular because of CRISPR, which you may have heard about. I was just looking at my phone while I was waiting to start, and I saw that Vertex had announced almost $1 billion stake for 10% of CRISPR therapeutics. So I'm thinking, dang, gene therapy is becoming lucrative, and I don't have shares in any of these companies. Okay, so this is just an overview of current gene therapies, not including cancer, that are currently in advanced clinical trials. They're on the whole run by companies, and the column on the right just shows you some of the different companies that are involved in this space. And highlighted in pink are the gene therapies that have received regulatory approval, either from the FDA or the EMA, which is the European equivalent of the FDA. So gene therapies really are now becoming credible medicines that you can prescribe for patients. I've already mentioned gene editing, so I'm going to give you a little bit of background because some of the slides later on talk about this. So gene editing is the ability to do very precise gene therapy, and it's mostly now being catalyzed by the discovery of CRISPR-Cas9. So for those of you who aren't sure what CRISPR-Cas9 is, I have a cartoon over here. Cas9 is an endonuclease protein. It makes cuts in DNA. And the CRISPR part is also called a guide RNA. And the guide RNA is synthesized to have homology to a DNA sequence you might, or a gene you might be interested to modify. And the guide RNA brings the Cas9 protein to that DNA target, and then it makes a very precise DNA break at that site. And CRISPR-Cas9 is kind of exciting because it's really cheap and easy to make these different guide RNAs that direct the Cas9 to different sites. And so in theory, you can make a CRISPR-Cas9 that can bind to and cut any sequence in the genome. Okay, so what happens after the CRISPR-Cas9 makes a DNA break? Well, then the magic of DNA repair is exploited by gene editors to create different outcomes. So on this slide, I'm showing here a cartoon of CRISPR-Cas9 making a DNA break. And then depending which pathway of DNA repair is used, this gives you different outcomes. So for example, in red, I'm showing that if a pathway called non-homologous end joining is used to bring together the two cut ends of the DNA, sometimes that happens in an imprecise way. So little bits of the gene are kind of lost at the joining site. It creates a sort of a scar, if you like. And that scar then disrupts the gene and leads to gene knockout. So this is a way to destroy a gene. Perhaps not terribly useful in a lot of cases. There's not many cases where you would want to knock out a gene. So of more use is the pathway shown on the right in green, which is called the homology-directed repair pathway. And here, the DNA break is repaired by copying a template that shows what the sequence should be corrected to. And in nature, the template is typically the sister chromatid. But we can hijack that pathway by introducing into the cell at the same time what we call a homology template. And this can be on any DNA molecule, for example, a plasmid, a single-stranded oligo, or a DNA viral vector. And then the genetic information contained in the homology template is used to repair the break. And if we put mutations into this homology template, or even additional DNA sequences, those also get copied during the repair process. And this therefore allows us to site specifically mutate a gene or add a gene at a specific place. So this is the power of gene editing. And so that's also really taken off. Again, just showing you an overview to sort of really highlight the breadth of applications that gene editing is now being applied to. A great deal of interest in cancer. You'll see that a lot of companies are working in the cancer space, but also hemoglobinopathies, beta thalassemia and sickle cell, as well as, of course, HIV. And I'll be touching on one of these clinical trials later on. Okay. So that's my kind of introduction to gene therapy. And so, of course, a bigger question, I suppose, is HIV disease a good candidate for gene therapy? And when I think about answering this question, I break it down into some of the characteristics of HIV, the virus, and how it infects and the damage that it does to convince people that I do think it's a good candidate for gene therapy. So let's walk through some of those characteristics. So first of all, HIV is a retrovirus, and therefore when it enters a cell, a CD4 T cell, for example, its genome, which is this squiggly line in hot pink, its genome integrates into a host chromosome, and the virus then becomes a permanent part of any cell that it infects, as well as any daughter cells that that cell produces. So it effectively becomes a part of our genetic material. And so I think of HIV as being like an infectious genetic disease. And as a result, it to me is sort of a no-brainer that the applications of gene therapy that are currently being applied towards genetic diseases, such as sickle cell disease, could also be applied towards this infectious genetic disease of HIV. The second major feature of HIV that you're probably all familiar with is HIV doesn't just integrate into cells, but after it integrates, occasionally it can become latent. Its genome switches off and the virus doesn't make any new, the infected cell doesn't make any new viruses. And one of the consequences of this is that although a productively infected cell can be seen by the immune system, it's making these foreign proteins, a latently infected cell is invisible to the immune system and looks, to all intents and purposes, exactly like a non-infected cell. So that's a problem because it means the immune system can't remove latently infected cells. The way we think they establish is it may be that HIV infects a T cell that is activated, but is transitioning to a more quiescent cell, such as a resting memory T cell. And as the cell is sort of shutting down, HIV also shuts down with it. And these latent viruses persist, including if the T cell expands, as T cell clones can do. So this is a way that latently infected cells are produced and are maintained. Now, if latently infected cells stayed latent, they wouldn't be a problem, but of course, they can be reactivated. And that means that even if just one latently infected cell reactivated, it could kind of restart a flare-up of the infection. And so the consequences of this are the sort of well-known rebound of viremia that's observed in patients who have been on antiretroviral therapy, but then stopped taking the drugs. And this is because, again, even just one latently infected cell rebounding can start the infection again. The third point I think of as one of the problems that HIV presents is that it's an incredibly mutable virus. Right now, everybody's kind of a bit focused with COVID. I'm sure you guys have all heard of the kind of concern about variants that are arising, the South African, the Brazilian, the UK variants of COVID. And these variants of COVID have like a couple of mutations in them, and it's taken, almost a year and, 130 million people infected for COVID to do this. HIV does this in a week, in a single person. It's a highly mutable virus. And as a result, the virus can, if you like, keep one step ahead of our adaptive immune responses, by which I mean both the antibodies that we produce that should recognize the outside of the virus, as well as the CD8 T cells, which are the cellular immunity that can kill infected cells. And so this is a problem. It means we are always trying to keep up with this virus. And then finally, and I want to spend a little bit of time on this slide because to me, this is kind of the really sort of central aspect of HIV. We all know that HIV infects CD4 T cells, but what you maybe haven't sort of thought through is the fact that by infecting CD4 T cells, and in particular, what we call CD4 helper T cells, HIV completely thwarts all of our immune responses against other infectious agents and, of course, against itself. So this is really the superpower that this virus has. So my cartoon shows an extremely simple overview of immunology, if you like. Down at the bottom, we have the two main types of adaptive immune responses, the B cells in blue, which secrete antibodies, and then the so-called CD8 T cells in purple, which have on their surface what's called the T cell receptor that acts a little bit like an antibody and allows them to bind to and detect HIV-infected cells and then kill them. And both B cells and CD8 T cells need what we call help from CD4 T cells to kind of be activated. So when HIV infects CD4 T cells, it can wipe out this help, and therefore both the B cells and the CD8 T cells don't do their job as well as they should do. And this is a really, central aspect of why HIV infection leads to profound immune suppression in the absence of antiretroviral therapy. Okay, so that's the sort of setting the scene, if you like. And for the rest of the talk, I'm going to highlight a couple of approaches using HIV gene therapy that are sort of caught my attention in the last 6 months or so. So first of all, just the sort of general types of approaches. I think of them in three different buckets. First of all, you can use gene therapy to protect a cell from HIV infection to make the cell resistant to HIV, and we'll talk about some examples of that. And then secondly, you can use gene therapy to improve our immune responses and enable the antibodies and the CD8 T cells to be better at attacking HIV, even despite all these things that HIV does to thwart those responses. Finally, and I'm not going to talk about it today just in the interest of time, but there's also interest in using gene therapy to remove these latent HIV viruses. And so I think that an eventual successful HIV gene therapy for an HIV cure is probably going to involve components of each of these three approaches. Okay, so first of all, let's look at protection and how you can make CD4 T cells resistant to HIV. So the classic and the first gene therapy approach for HIV was based on knockout of the CCR5 gene. So why was this the first target? Well, CCR5, which is shown in this cartoon over here, is the major entry co-receptor for HIV. HIV viruses, when binding and infecting a CD4 T cell, have to bind to both CD4 on the surface of the cell, but then also the GP120 protein here in orange also needs to engage with a so-called co-receptor, and CCR5 is the dominant co-receptor that HIV uses. What's interesting is that the CCR5 gene actually isn't essential, and people can live quite happy, completely normal lives without CCR5. But if you have 2 copies of a mutant form of CCR5 called CCR5 delta 32, you don't have CCR5 on your cell surface, and therefore you are highly resistant to HIV. And as a result of this, transplantations of hematopoietic stem cells, which are the stem cells that produce all of our immune system and live in the bone marrow, so you can also call them bone marrow transplants, HSC transplants from CCR5 delta 32 donors are the only known cases of HIV cure to date. That's shown on this slide. There have been potentially 3 people who've been cured of HIV following a transplant from a CCR5 delta 32 donor. First of all, the so-called Berlin patient, Timothy Ray Brown, that I think many of you probably know. Sadly, Timothy passed this past September due to a recurrence of his cancer. Subsequently, we also have a new patient, Adam, who's referred to as the London patient, who also appears to be completely cured of HIV as a result of having this transplant. And there's also, although I haven't seen this yet published, evidence that there is another patient in Dusseldorf. Both Adam and the Dusseldorf patients were transplanted in the same way as Timothy, based on a consortium in Europe called ICSTEM. So let's take a look at what happened with Timothy and Adam, and that's shown in this cartoon here. They received a stem cell transplant from a donor, and as a result, they themselves became CCR5 negative and therefore were able to suppress HIV. Doing bone marrow transplants is incredibly difficult and actually can have quite a high degree of morbidity and mortality. So unless you have an underlying blood cancer, this is not going to be an approach. So instead, what gene therapists do is they take the stem cells out of the HIV positive individual, and in the lab, using the tools of gene editing, they are able to make at least some of those stem cells CCR5 negative and mimic the phenotype of the CCR5 delta 32 mutation before returning the stem cells to the patient. And down at the bottom here, I just have the cartoon to remind you that if CRISPR-Cas9 cuts in the CCR5 gene, the error-prone repair can lead to disruption of that gene. Over on the right, I've summarized various different ways that this CCR5 gene editing is being used. At the top is the IC-STEM consortium, which is trying to recreate Timothy and Adam's cure by identifying HIV positive individuals who also have an underlying blood cancer and therefore need a bone marrow transplant, and trying to match them up with donors who are not just a tissue type match, but also have the delta 32 mutation. And this is typically about 1% of the population. So it's not an easy thing to do, and you need large databases of donors to screen to find these potential donors. The autologous gene therapy trials, which is using a patient's own either T cells or hematopoietic stem cells. There's a number of these trials ongoing. In particular, they use not CRISPR-Cas9, but what are called zinc finger nucleases, which are like an earlier version of gene editing before CRISPR-Cas9. I'm going to show you in the next couple of slides some data from a clinical trial happening in Los Angeles at City of Hope that I was involved in doing the preclinical studies for. And then finally at the bottom, in a sort of a combination approach, there's a group in Beijing who are taking again individuals who need a bone marrow transplant because of an underlying blood cancer. And they are editing with CRISPR the hematopoietic stem cells from the donor to make the donor's cells CCR5 negative before transplanting them. So various different approaches. So as an update on the City of Hope trial, I have a couple of slides on that. First of all, this is just the details of the trial. It's a pilot study, phase 1, to evaluate the feasibility and safety and the engraftment of ZFN modified, CCR5 modified hematopoietic stem cells. And this is the information about the PIs and the various people involved. And down here is the clinicaltrials.gov identifier. This is a schematic of what the trial involves. I think of it in 3 different phases. The first phase is where the hematopoietic stem cells are collected from the individual after injection with what are called mobilization drugs, and then 2 rounds of apheresis. This takes about a week. Then the cell manufacture and editing phase takes about 6 weeks, where in this case, zinc finger nucleases are introduced and all the sort of checks for quality are performed. So the cells are frozen down during this period. And then finally, the actual treatment phase, the individual is treated with a drug called busulfan, which is like a mild chemotherapy, which makes space in the bone marrow so that the reinfused edited hematopoietic stem cells have a better chance of re-engrafting. This was done in 2 cohorts with different amounts of the drug. The cells are reinfused. And then, of course, there's a long-term follow-up, currently planned up to 15 years, to see if any of the CCR5 edited cells engraft and if they are maintained. In order to see if there is any effect against HIV, an analytic treatment interruption is planned at about 9 months. An ATI, again, I think you guys may be familiar with this. It's frequently proposed for HIV cure trials. This is the idea that you will temporarily withdraw antiretroviral drugs and look to see if the treatment has had any impact on the expected viral rebound. And so if the therapy has worked 100%, you could stop the drugs and the virus would not come back. much more likely to, what you would expect to see is that the virus still comes back, but maybe it comes back delayed or at a lower level, and that would be some indication of efficacy. ATIs have eligibility criteria that are based on the overall health of the individual after the conditioning and infusion, and importantly that there is at least some detectable CCR5 modified CD4 T cells in the blood before this would be attempted. So this is the data that City of Hope have presented publicly at this stage. They've treated 8 individuals. 2 individuals have been on ATIs, although I don't have that data. On the left here, excuse me, is just showing the levels of CCR5 disruption that were achieved in the stem cells in the laboratory, and they range from a high of 43% of the CCR5 genes were disrupted to a low of 8%. So you'll see quite a bit of variation patient to patient. And then over on the right, what you're seeing here is over time, how the levels of CCR5 edited cells that could be detected in the blood changed from an initial peak in the day immediately post transfusion to a more long-term steady state level. And you'll see that the levels are pretty low. This is sort of 1%. This individual has 3%. He or she is higher. And so the levels are pretty modest, although you can see that they trend. The orange levels are higher, and these are from the cohort that got higher levels of the busulfan conditioning, so implying that this conditioning is needed to help re-engraftment of the modified cells. Okay, so those studies are ongoing. Okay, so what I'm going to do now is switch to describing some applications of how we can enhance the immune cells involved in potentially controlling HIV, and I'm going to describe a couple of different approaches that have been pursued to do that, including some recent work out of my own lab. So the first thing I want to talk about is different ways of preserving T cell help. If you remember, we talked about the fact that the CD4 T cells help the CD8 T cells to recognize and kill an HIV infected cell. So there is a company called American Gene Technologies that have an interesting approach, and up on the top here is the clinical trials identifier, because their clinical trial started enrolling about 6 months ago. And so what they do is they isolate CD4 T cells from an HIV positive individual that are already programmed to recognize HIV proteins, and in particular, the HIV GAG proteins. And these are present, they're pretty rare, but they can purify them. And then what they do is they massively expand these HIV-specific CD4 helper T cells in the lab by stimulating them with peptides from HIV GAG. And at the same time as they are expanding them, they also use gene therapy. In this case, it's with a lentiviral vector to make the cells resistant to HIV. And the lentiviral vector contains shRNAs against both the CCR5 protein, mimicking the CCR5 knockdown we were talking about with gene editing, as well as having some shRNAs that specifically target HIV proteins. And so after the expansion, they basically go from 1 million cells to 1 billion cells, and then these get reinfused back into the individual. So it's kind of really boosting the body's natural CD4 helper T cells that could recognize and help HIV-specific CD8 T cells. A slightly different way of doing this is, rather than focusing on the CD4 helper T cells, instead to focus on the CD8 T cells themselves and make them specific for HIV. So to do this, HIV is taking a leaf out of the cancer cell therapy book. You guys may have heard of CAR T cells. CAR stands for chimeric antigen receptor, and there's a lot of excitement about CAR T cells, especially up in the Seattle area where a lot of this technology was first developed and is being promoted by a company called Juno. And the idea here is that, whoops, let me go back, that normally a CD8 T cell can recognize a tumor cell as foreign because a peptide from that tumor cell is presented to the T cell receptor. And so the chimeric, the CAR is basically an artificial T cell receptor that is made to recognize a target on your tumor cell. And in B cell, these are normally done for B cell tumors where the target is something like CD19. So to make the chimeric antigen receptor, oftentimes the tip of it is part of an antibody, a single chain antibody. And so if you wanted to target an HIV infected cell, and in particular the GP120 protein, which will be on the present on the surface of a HIV infected cell, you can take an antibody that recognizes GP120. But you can also take part of the CD4, the extracellular domain of the CD4 protein itself, because this is what the HIV GP120 protein normally binds to. So this is, if you like, a perfect receptor for GP120. So the way that this works, for example, I'm showing you here the approach of a company called Lentigen. And again, here's the clinical trial identifier. And they started a clinical trial right at the end of 2020 that's been run out of UC San Francisco. And they have what they call a duo CAR approach. They introduce two different CARs into a cell, into the CD8 T cell. This is shown here by this squiggly yellow line. And so the CD8 T cell now has two different CARs on the cell surface, both of which can interact with GP120 on the surface of an infected CD4 positive T cell. And their CARs recognize two different parts of GP120. And these are these two different domains shown here. And so the way that this works is very analogous to what I showed you previously with the hematopoietic stem cells, except this time the T cells are isolated from an HIV positive individual, engineered ex vivo with the lentiviral vector expressing both of these CARs and then reinfused. And I also have a reference down here describing the underlying technology. And then finally, just to kind of blow everybody's mind, I was really struck by this, this study from a few months ago from Jim Riley's group at UPenn. The UPenn team have really been ahead of everybody, I think, in developing CAR T cells, both for cancer and for HIV. And what Jim's group is doing here is they are combining both putting a CAR on CD8 T cells, which is this cell over here, allowing it to recognize an HIV infected cell. But at the same time, they're also adding their CAR into CD4 T cells. And to prevent the CD4 T cell being infected by HIV, they also add, in this case, it's an entry inhibitor using gene therapy. And what this sort of means is that they can really get everything kind of together and all on the same page. So your HIV infected CD4 T cell is going to attract both the CD8 CAR T cell that could kill it, but also the CD4 T helper cell, which is providing this T cell help. For example, the cytokines IL-2 and IL-21. And the idea is that this can really stimulate and enhance the killing of these HIV infected cells. And they found, and here's the paper down here, that having both engineered CD4 cells and CD8 cells went a long way to really improving the ability of the CD8 cells to kill the HIV infected cells in a humanized mouse model of HIV. Okay, so for the last part of the talk, I'm going to tell you about work that my lab does, where we are instead working on the B cells that produce the antibodies and thinking if we could enhance that process. So I need to introduce you to something called broadly neutralizing antibodies. So one of the challenges with HIV is that although HIV infected individuals make perfectly good antibodies against HIV, HIV, because it keeps mutating, is able to very easily mutate away from those antibodies. However, rarely, in particular in people who've been infected for a long period of time without antiretroviral therapies, they can develop special types of antibodies that we call broadly neutralizing antibodies. And these have strange properties, but oftentimes they've got, if you like, long fingers, and they can dip down into highly conserved regions of the HIV envelope protein that can't easily mutate. It's kind of like a checkmate move. And so broadly neutralizing antibodies, you know, there's a great deal of interest in them. Unfortunately, because they are so rare and so unusual and so highly evolved, they can't easily be made by vaccination. So, you know, we haven't been able to find a way to immunize people that results in them making broadly neutralizing antibodies. And so instead, the way that we use broadly neutralizing antibodies currently is they are injected as monoclonal antibodies, recombinant proteins, and they work, but you have to obviously keep giving people injections, and it's, you know, that's a pain and it's expensive. And so gene therapists are also thinking about, can we use gene therapy vectors that carry the information to code for these broadly neutralizing antibodies and inject them, for example, into the liver or into muscle, and the body then has a factory inside that can be making these antibodies. And although that looks like, you know, a promising approach, making antibodies from non-natural tissues such as liver or muscle often creates, ironically, antibodies to the antibodies because they're kind of a little bit foreign. So instead, my group has been working on whether or not we can make these broadly neutralizing antibodies from the natural cells that make antibodies, which of course are the B cells. So just a bit of a pitch for B cells. I really do think that antibodies naturally produced from a B cell are a perfect drug. First of all, B cells, in this case, memory B cells that are they express the antibody on the cell surface. It's called a B cell receptor in this format. When they encounter their antigen, in this case, it would be HIV, they become stimulated, they expand, and they start to secrete antibodies. And so you can boost antibody production, for example, in the presence of an infection or by vaccination. So they are boostable. The expanded B cells eventually form what are called plasma cells, which are these long-term factories in the body that secrete antibodies. They can do this for upwards of a decade. So they're a great cell to be making the antibody. And then finally, antibodies also have another trick, which is that they can undergo mutation and can be enhanced and matured during a process called affinity maturation, so that even if your antigen or your virus starts to mutate a little bit to try and get away from the antibody, in theory, antibodies can evolve to kind of try and keep pace with that antigen. So the approach my lab took is that we wanted to engineer B cells. But when we looked at antibodies, and on the left here is a natural antibody, and it comprises two chains, a heavy chain and a light chain. So that's 2 different genes you would have to do gene editing in. And we just thought, gosh, that's just way too complicated. So instead, we mimicked an approach of what are called heavy chain only antibodies. And an example is shown here. And these are natural antibodies that are found in camels and llamas. You know, who knows why they developed this, but it's a cute trick. And the antigen recognizing tip of the antibody, which is called the VHH cassette. This is a single fragment. It's very compact and it forms the basis of a technology called nanobodies, which are increasingly being used as an alternative to traditional monoclonal antibodies. Okay, so my next slide is probably the most complicated slide of the talk. So, you know, if you need to sip your coffee at this point, that's an okay time to do it. I don't really have the time to go through this in detail, but I'm going to try and give you the high level of how we use gene editing to turn human B cells into cells that can secrete human heavy chain only antibodies. So first of all, this is just a very simplified version of the natural heavy chain gene in human cells, which has the different components that together give rise to at least the heavy chain part of a conventional antibody. We use CRISPR-Cas9 gene editing to make a DNA break at this point and insert in a cassette brought in by a homology donor. And the end result is that the edited locus has this cassette inserted upstream of the constant region of a traditional heavy chain. And as a result, we can produce both secreted heavy chain only antibodies as well as subsequent to differential splicing, the membrane bound form of the antibody, which is called the B cell receptor. Does this work? Yes, it does. Again, a slide with just some data showing you this, that our engineered human B cells, first of all, we can detect that they make, and this is, we've engineered them with broadly neutralizing heavy chain antibodies. We can detect this on the surface of the cells. If we take B cells, human B cells that we've edited, and then we differentiate them in the lab towards plasma cells, these antibody secreting cells, we see that what happens is the form of the antibody changes from being the membrane bound B cell receptor to the secreted antibody, as we'd expect. And then finally, if we take these secreted antibodies and add them to a HIV neutralization assay, we can see that as we increase the amount of the antibody, we can block HIV infection, so they retain full functionality. So my lab is actively working on this. We think that engineering B cells is kind of like a nice complement to the work that's been done by many other groups engineering T cells, and will give us some of these different options to target HIV. So to summarize, first of all, a summary about gene therapy. So I hope I've kind of convinced you that gene therapy is increasingly being used for diseases, especially those with few or no other options. And the FDA has currently approved 4 gene therapies, but by the last count, more than 900 are in development. So I think we're really going to start to see an explosion of these therapies. Advances such as CRISPR-Cas9 and gene editing, which allows much more precision in gene therapy, is really expanding the options and creating a lot of excitement. And then this is an interesting and maybe controversial point, but public awareness, I mean, people have heard of CRISPR, and possibly acceptance is growing. And this is helped by the success of current clinical trials for things like the severe combined immune deficiencies, sickle cell disease, cancer, and blindness. And again, I told you, I just saw this news item this morning that Vertex has put nearly $1 billion into CRISPR therapeutics for a small stake in the company. So it's also, possibly a lucrative sector of biotech as well. And then interestingly, I think with all the discussion about the COVID vaccines, the RNA vaccines from Pfizer and Moderna, which are a type of gene therapy, and the adenoviral vector vaccines. I find people are increasingly interested in the idea of gene therapy in general. So you know, maybe a silver lining of the pandemic. And then the summary for the HIV gene therapy approaches I've described. First of all, the way that we can protect CD4 T cells to make them resistant to HIV, for example, by knocking out CCR5. And then various ways that we can go on the attack, enhancing immune responses by expanding and protecting CD4 T cell help, by engineering CAR T cells to improve the CD8 T cells, and potentially by engineering B cells to express these hard to generate broadly neutralizing antibodies. Whoops. And then I didn't have time to talk about this, but there are groups that are also working on in vivo delivery of CRISPR-Cas9 that recognizes and could destroy the integrated latent HIV. And I will just end with an acknowledgement of of my group, who I haven't seen for over a year, but I believe they are still turning up in my lab, and the funding that supported the B cell studies. Okay, and with that, I'm happy to take any questions you guys might have.
Okay, so we've got a two-pronged question here. Can you talk about the costs associated with gene therapy cures?
Gosh,
and
yeah.
and the ability for us to do gene therapy in places in the US and around the world where HIV is most prevalent?
Okay, those are great questions. I think I'm going to stop sharing the screen. So first of all, the cost. So the whole field of gene therapy is grappling with what should you charge, because these are not conventional drugs at all, and a lot of this is being worked out, especially with CAR T cells for cancer. The current costs are astronomical. Some of these gene therapies are $1 million a treatment, and I think over time a couple of different models are going to be considered. One model might be a sort of like a subscription model, if you like, whereby the healthcare provider, pays a sort of a licensing fee as long as the gene therapy is still effective. Maybe a yearly payment, and those prices are typically pegged to be the kind of equivalent cost of the current therapy. So if antiretrovirals cost a certain amount, or if the consequences of a disease incur certain charges, then that would be considered an appropriate starting point for discussions about these therapies. A complicating factor is, of course, that these are not currently not just drugs that you can inject, get off the shelf and inject into people. They are, at this stage, highly personalized treatments. And so as a result, there is a lot of complexity and expense that goes into preparing them. And this is kind of leading into an answer for the second question about, what can be done to make this more accessible and available, both within the United States, and I would also say globally, especially in sub-Saharan Africa. So people are thinking, how can we make, gene therapy just, easier to do? A couple of approaches. One is we're seeing companies developing what's called sort of gene therapy in a box, so that instead of the current process whereby you take cells out of somebody, they are treated and expanded in a lab. You remember the slide I showed you from the City of Hope trial that had the very complex process, needs clean rooms, needs, highly qualified technical staff to do this. Instead, in theory, what you might have is a box that in your fantasy could be in a clinic, and people go in and it takes out some blood and they goes through a machine, and the machine does all the steps of the gene therapy, isolates out the cells you want to do the gene therapy on, introduces the gene therapy, and then produces a product that can then be reinfused into the patient. So I think there's a lot of excitement about the idea of these sort of, almost like mass production machines that can be in clinics to do this. The holy grail of what we really need that will be the game changer is to have ways to do gene therapy, not by taking cells out of the body and having to engineer them, but instead by having a gene therapy that can be injected into somebody, for example, in some sort of smart delivery system that goes to the T cells in the body or goes to the hematopoietic stem cells in the body and does the gene therapy in the body. And if that was possible and was safe, that would be a real game changer, because then you are turning gene therapy into a drug rather than the current complex medical procedure. Of interest, I'm doing two things at the moment. I'm also supposed to be chairing a workshop at the National Institutes of Health, which is looking at a 5-year plan to do exactly this. How do we get gene therapy and gene editing in particular into the realm of in vivo injectable drugs that can do gene editing in the body? So although it sounds a little bit like science fiction at the moment, there is funding and a great deal of interest in doing this. And certainly NIH are putting quite a lot of money into this goal. Sorry, very long answer, but you did give me 2 questions.
Okay, do you think CRISPR would be a way to target cccDNA in hepatitis B infections?
Yes, absolutely, yes. And in fact, my friend Keith Jerome at UW is working on that, I understand. In a way, any chronic viral infection where we know what the viral genome is could be a target for a CRISPR therapeutic. Again, what you would need is a delivery system that went specifically to the cells that were infected. Maybe not quite so difficult as the HIV challenge, because it's relatively easy to deliver things to liver cells. So I think that's actually quite a promising approach. In contrast, I probably didn't stress this, but when we're thinking about delivering CRISPR, for example, to disrupt an integrated latent HIV genome, we are looking for a needle in a haystack. Only about 1 in a million quiescent memory CD4 T cells in the body of somebody who's HIV positive harbors a latent integrated HIV genome that could potentially wake up and restart an infection. And because the virus is latent, it's not expressing any HIV GP120 on the cell surface. It looks just like a completely non-infected cell. So how do we deliver CRISPR-Cas9 to those cells that have the integrated HIV? And ideally, we would want to only deliver to cells with the integrated HIV. And at the moment, that's a huge problem that I haven't seen any good solutions to. But people are kind of excited about the potential for CRISPR to disable at least some of the integrated viruses and reduce the burden of the latent HIV reservoir. And there's a company called Excision Biotherapeutics that are doing this work, and it'll be interesting to see what progress they can make in the next few years.
All right, we've got time for a couple more. Question regarding your engineered B cells.
Mm-hmm.
Do you anticipate the secreted antibodies will neutralize cell-free virus? Or do you expect them to kill HIV-infected, latency reversed cells by ADCC or both?
That's a great question, both. So antibodies, when I was, before I had to study them, I used to think antibodies just bound to virus and neutralized it. They sort of covered the virus and stopped it infecting cells. But antibodies do so much more than that. They can also recognize a virally infected cell and bind to the GP120 on the surface of the cell. And then, antibodies are this sort of Y shape, and the sort of the stalk of the antibody also has functions, and it can bring in other activities, including, bringing in cells that can eat, phagocytose the infected cell. And what's increasingly exciting, I think, in this area is that you can also engineer antibodies so that the stalk part has enhanced functions that really promotes this so-called ADCC cell killing, or ADCP, I think it is, which is the phagocytosis. So in our antibody engineering strategies, as well as replacing the, if you like, the head of the antibody, so that it now recognizes HIV, we are also replacing the stalk or mutating the stalk, so it has these enhanced functions. So absolutely, we would be aiming for an absolute, supercharged antibody, better than nature, that did all of these things. Yeah, that was a great question.
All right, last question. Would a broadly neutralizing antibody product be effective in any patient and effective against most HIV strains,
Right.
i.e. not autologous and lower cost?
Yeah, so that's the hope with broadly neutralizing antibodies. They are broadly neutralizing, meaning, they do recognize multiple different strains, including from quite diverse different subtypes of HIV that you're getting different parts of the world. Of course, there is always the challenge that if you make a chess move against HIV, it makes a chess move back against you. So if you give individuals just one broadly neutralizing antibody, even if initially it neutralizes their virus, the virus quite quickly evolves resistance against that. So broadly neutralizing antibodies will probably also be deployed in combinations. And in our B-cell engineering strategy, we also have found that we can sort of daisy chain different antibodies together and make these sort of linked broadly neutralizing antibodies that present much more of a challenge to the virus. So it's a little bit like, combination antiretroviral therapy. But so that's probably how we're going to have to use these antibodies.
Thank you so much, Dr. Cannon, for this excellent talk. There were some comments about how it was such an excellent talk and a really wonderful explanation of very complex thoughts and ideas. So thank you.
It was complex. My apologies.
No, but I think your explanations were very clear and your illustrations were very clear. So it was really helpful for lay people to even understand this complex
Thank you.
field of study. So thank you so much.
Okay. All right. Well, goodbye, everybody. Thanks for your attention. And please, if you have any questions, feel free to email me. I occasionally respond. And enjoy the rest of your day. I'm going to go get coffee now before I pop back into my other meeting. So.